one.
They don't need to hear me anyway. By the time I'm done, it will be.
Put out my next thing.
So it is a real treat today to have Henri back on the occasion of his events and to have his family back too. And all of you know Henri, so I really don't actually need to introduce him. But well, probably most of you don't know the full history. So Henri is obviously from South Africa. He did a bachelor's degree in nature conservation at Nelson Mandela Metropolitan University, which is in the city formerly known as Port Elizabeth. And I haven't learned the new name yet, which I need to do. But he then went on to a master's in environmental management at the University of the Free State. And eventually he joined the Buffalo project that we ran in South Africa between 2014 and 17, which followed a buffalo herd that was captured in a really large enclosure for 3 years. And Henri was very instrumental in managing that study. And he's always, at least since I've known him, had a really keen interest in spatial processes and spatial patterns in ecology and in animal behavior. And weirdly enough, it ended up that the data that he ended up using for his PhD actually came from the previous Buffalo project, because that project followed a bunch of free-ranging animals that have more leeway in how they make their space use decisions than the animals that Henri actually was involved in the fieldwork with that were in this large enclosure. And so he actually did a ton of work analyzing data from the previous project that he'll talk to you about today. But aside from just sort of the the academic bits and pieces, are you wanting to come in, please? Thanks. Thank you. I always like to at the time of the event sort of reflect back on what are the students' superpowers really, and everybody has them and they're all different. It's kind of interesting to think about. And Henri has plenty of superpowers, and I probably know more of them because I've also been his neighbor and his friend and his running buddy, as well as his advisor. And so I'll just mention 3 of them that sort of sum things up a little bit. The first one is that Henri's probably the most independent learner I've ever not taught. He didn't need to be really taught in anything because he tends to teach himself, and it really doesn't matter what it is. So if you're trying to set up an irrigation system in the middle of an African savanna, well, Henri's going to figure it out and do it. If the stats that are available commonly for a particular question that he gets interested in, and when he's interested, he's really interested. If those don't really exist yet, well, he'll find some stats from some other field and sort of transmogrify them and learn them and apply them to this new problem. If the engine breaks on his car and it's a bit expensive to fix, he's just going to do it himself. So he's an incredibly independent-minded and capable person, and particularly this is the second superpower. He has a very keen mind for analysis and for numbers and for stats, and he's really pushed what we as a group have done with large complex observational data sets a step beyond what we've been doing previously, and he'll show you some of that work today. But what's kind of funny is that with that keen quantitative mind comes really sort of almost a bit of boredom when it's just a main effect. Main effects are just kind of a little dull to Henri. And it gets just that much more exciting when you can have like some model predicted parameters, and then maybe you do a data reduction step on those, and then you can maybe represent those visually in some graphs that have at least 3 different variables that are moving with colors and axes and stuff. And then you have some, multivariate interactions, and the interactions are where it really gets exciting. So Henri is like, usually 3 steps removed from really dull main effects. And so then the rest of us need to catch up to that. And then the final superpower that I'm going to mention here before I hand over the room to Henri is that he did this tremendous amount of work. It doesn't all very easily fit in a defense seminar, you'll see. But he did that also while raising 2 extremely boisterous girls, real energy bundles. And I have watched that because I've been my neighbors bouncing on my trampoline, and we've been at each other's parties, and Henri's really just an amazing dad. And I think, you know, balancing that as well as producing this academic work definitely qualifies as a superpower. You know, he's like a more quiet person, and so when there's all this chaos going down, he's the rock, and he manages with, you know, keeping kindness in his heart when when they're being ridiculous and dramatic. He actually keeps it straight. It's amazing. And he's the dad that empowers his little girls. By the time they were 4 years old, they were like, oh yeah, I can take my backpack and hike in 10 miles to a beautiful lake. And then it rained and the tent was very cozy. That's Henri. So with that, I'll hand over to you.
Thank you, Anna, for very kind words. And welcome, everyone. So today I'm going to take you through a very like a bit of a whirlwind tour through some aspects of my thesis entitled Navigating Food and Flows in Savannah. So we're going to be looking at some nutrition, infections, and how they interact in the landscape in Africa. So to start with, a central goal of ecology is understanding the patterns of organismal diversity and abundance. Let me start that time again. Organismal abundance in both space and time. So this can range from megafaunas, as my study species, African buffalo, all the way down to, you know, microorganisms and parasites. So predicting the temporal and spatial patterns of the occurrence of these infectious organisms is the purview of epidemiology, and really what we are interested in. And this is really relevant, and as we all know from recent events, we're up to about 75% of recent zoonotic diseases are known to have animal origins. So it's therefore essential that we develop strategies that to prevent and control the spread. So if we then zoom into the African buffalo, my study species, this is a really good example. African buffalo are a species of economic importance. They're charismatic species. They one of the big five species. They form a big part of the ecotourism industry in South Africa, as well as a really large private game industry with people breed them for the big horns, and then they get hunters lots of money to go and hunt them. But on the flip side, these animals play host to a lot of diseases and parasites that become endemic within these natural systems, and most of these has either demonstrated already or has a zoonotic potential. Here's just a couple of examples. For instance, over here I have foot and mouth lesions on the tongue of a cow there, and foot and mouth disease was introduced into the system, it's become naturalized from livestock, and now it's actually a problem with neighboring communities and livestock around conservation areas where we need to manage these diseases. On the right here, for instance, TB, which is also an introduced disease into these natural systems. So you can see some nodules on the lungs there, which is also a disease that we need to manage and that kind of contain within these natural conservation areas and prevent from spilling over into livestock and causing economic damage. And then we also have various tick-borne diseases, which we can also spill over into domestic livestock. So a real challenge is that these diseases become naturalized in the ecosystems. So therefore, we need to implement effective management strategies to manage it. And it's essential to understand both the spatial and temporal dynamics for us to achieve this outcome. So if we think about managing diseases, and especially if we think about epidemiology, we always refer back to the paradigm of the epidemiological triangle, or the epi-triad. So the epi-triad basically looks at the parasites and how they interact with the host, and then how on the backdrop which enables all of this to happen in the environment. So for my thesis today, I'm basically going to use the epi-triad as a roadmap to go through some aspects of my dissertation. So we're going to start off looking at parasites, and we look at how parasites are distributed in the landscape and how that affects exposure risk to animals for parasites and pathogens. Then we're going to zoom into the environment, and we're going to look at how nutritional heterogeneity or heterogeneity access to nutrition affects the life history outcome of African buffalo. And then we're going to conclude and zoom into a specific example, being Schistosome matei. We're going to look at variation resistance and how that affects the life history outcome of these animals. So as Anna mentioned, this was a large study that happened over 4 years. So I'm just going to give you a brief background to the study. So the study occurred in South Africa in the Kruger National Park. Kruger National Park is about 2 million hectares in size, it's roughly about the size of Israel. So it's a long, narrow conservation area on the boundary with Mozambique and Zimbabwe. We focused on the south central area of the park, where we conducted all the captures of the buffalo that was involved in this study. This is just a map to illustrate the buffalo population distributions within the park. So buffalo occur throughout the park, but they're bound to very specific habitats that are suitable for them. Buffalo, at the turn of the last century, the populations were really small due to hunting pressures as well as the rinderpest economic, but the buffalo population has grown today to around about 30,000 animals in the park. So when I go through my results today, I really keep it in the back of your mind that these systems are highly dynamic systems with a lot of spatial variability that drives all the dynamics that I'm going to be discussing today. So for instance, here's a soil map that I just generated. So you can see over a short amount of space, there's a diversity of soil types that influences what will grow on there, what the nutritional quality of the vegetation points will be. There's a great diversity in the average rainfall, primarily driven by orographic effects from nearby mountains, as well as the local relief. So you can see here over a short distance here, you also get quite a diversity of rainfall. And especially sometimes you can have rainfall in this part and 2 or 3 kilometers away, it won't rain for 6 months. Generally, there's a rainfall gradient, so more rainfall in the south, and it generally gets drier as you go up to the northern plains. And this all then starts translating to diversity of the vegetation and the habitat that's available to buffaloes. Here's a woody cover map just to illustrate the diversity that you get in tree grass interactions. These are also fire-driven systems. So fire really drives the dynamics of this vegetation. It drives what the tree grass ratio is in the savanna mosaic and how much forage is available to buffalo. And this map is just to illustrate, this is a satellite image of different age fire scars that you can see in the landscape. And then also the central area here got burnt here in what's known as a firestorm event. So that's where you get really hot burns that basically kill a lot of trees. And these are also play a vital in the dynamics of species, because they often cause like green flushes when it rains. Animals will congregate on there, and that'll be a mixing bowl for disease transmission as well as dispersal events and things like that. And next we have a great seasonal variability as well. So this mosaic here on the left is pictures I took from one marula tree over a couple of seasons, and you can see in the wet season here it's very lush. All the trees have leaves on them. The grass grows really tall, right about up to my height, and they're very palatable and nutritious, and the animal can gain weight and gain body condition and do really well. Then going into the dry season, the trees lose the leaves, the grass becomes senescent and become very unpalatable, and animal will lose condition during those times. This over here on the right hand side, a similar story. It shows more representative of the north central area of the study area. We can also see nice and lush during the late wet season to very dry conditions during the late dry season. So this all translates into variable forage quality for animals. So here on the x-axis is the time throughout the study period, and on the y-axis is a dietary called fecal chlorophyll, which we'll be seeing later in my presentation today as well. But it's just to illustrate that animals' vegetation quality declines, the green forage that's available to animals declines, then rains again, and it cycles like that throughout the study period. And then this all translates into nutritional variability for animals. So this is just dedicating time on the x-axis. On the y-axis is a percentage, and the bars is the percentage of animals that have caught that was nitrogen deficient. So that is nitrogen, dietary nitrogen under 1.2%, we consider nitrogen deficient. And that then cycles asynchronously with body condition and the general greenness within the landscape. Okay, so there's long periods where animals are nutritionally not doing very well in the system. Then also most ungulates in African savannas are herd-dwelling animals, and especially in buffalo, and I'll speak about this a little bit again later. It's really strong static social dynamics, which causes a great disparity in terms of forage quality that's accessible to individual animals. So just very briefly, you can see, for instance, here, there's a little grouping there. Then there's animals here in the front, Those are normally more dominant animals that gets better quality forage. And then you have these stragglers here in the back that are normally older or subdominant animals, which then don't get access to good quality forage compared to the individuals in the front, although they're grazing on the same vegetation. And then lastly, this is a complex risk landscape with buffalo infected by a suite of micro and intestinal parasites. So we have schistosomes, we have nematodes, we have coccidia, we have ticks, and then the vector to a whole host of microparasites. And then this, on top of all this, animals also need to navigate predation as well. And that all influences where they eat in the landscape and the risks they have to navigate to obtain adequate nutrition. So as I mentioned, this was a large project. So the backdrop of this project was that we were interested in understanding the interaction between nematode intestinal parasites and tuberculosis, bovine tuberculosis dynamics. So half of this population was inoculated with the Hispanic herpetosis, which suppressed the nematode population, and then we were interested in seeing how TB reacted to that. So like Anna mentioned, I did a lot of field work, but the people who actually collected most of the data that's included in my PhD was collected by the people you see on the pictures here. So thank you to them. Without them, I would not be standing here today. So as I mentioned, these animals were captured. Around about 200 of them were captured initially via helicopter darting. So they were immobilized, and then some of them received collars, they received tags, so we know who they are. And then these animals were followed up every 6 months via vehicle-based darting techniques. So that resulted in about 261 animals being darted in total. So dead animals were replaced to keep roughly about 200 animals in the study. And that resulted in about 1,326 samples in total. And that's about 2 to 9 samples per animal, depending on how long the animal lived in the study period. So just briefly, what did we collect when animals were down? Age was collected by estimated by incisor immersion techniques. We collected horn width. So here we're measuring the width between the widest points of the horns, and that's a metric I used to assess investment in somatic growth. So how much energy does an animal put into a secondary sexual trait? We assessed the body condition here. So we palpated 5 areas on the buffalo that's indicated by the arrows here. We then averaged that, and that's a qualitative score of body condition. And then lastly, we assessed reproductive status through rectal palpation to basically feel if there's a fetus there. And you also determine if animals were lactating and whether a calf was present or not. These are strong dynamics for life-century outcomes, but also it affects a lot of the diseases as well that we're interested in. And then we collected bloods, which we took back to the lab, and we collected a whole host or suite of disease and parasite diagnostics, which all is there. And as well as a lot of immunological measurements. So we collected everything from intestinal parasites to microparasites like bovine tuberculosis or parainfluenza virus, for instance. And the immune variables vary from like just basic blood cell counts to non-specific markers of inflammation to bacterial killing assays, things like that. So we have a whole host of measures that we looked at. I'll talk about mortality. So for mortality, we didn't exactly know when animals died, but we estimated that when animals were not captured for more than one sampling interval, and then we considered that the mortality interval. So that's what I'm talking about when I'm talking about the mortality interval. Later on, I'm also going to talk about dispersal a little bit, as well as the dispersing interval. So we considered an individual that was recorded outside of their herd of origins, home range, and that was not joined with another herd, and not returning to their own original herd for at least one season as dispersed to another herd. And the last sampling interval for that, where this event occurred, was considered the dispersing interval. And then lastly, we collected various nutritional metrics. So we looked at fecal nitrogen, which is an established metric of dietary composition, and basically it measures the percentage of dietary nitrogen in feces. And we also, around each capture point, so where we darted that animal, we estimated NBI from MODIS satellite images. So that's the normalized difference vegetation index, which just gives us a measure of the general greenness and the primary productivity of the landscape around that point. And then lastly, we used a relatively new novel diageometry called fecal chlorophyll, which basically just measures the chlorophyll pigment within a fecal sample, and we measure that at 606 nanometers, and that just gives us a metric of individual selection for active primary production. And this is just to show that all these metrics were nicely correlated. Okay, so that takes me to the first chapter that I'm going to be discussing. So here we're going to be looking at the distribution of parasites within the landscape and how nutrition, immunity, and these pathogen distributions are correlated with each other. So to determine disease risk for wildlife, we need to know about the spatial patterns, so where in the landscape are disease, as well as the temporal patterns, when the diseases occur and why. So it's important because it affects the number of susceptible individuals in the area, which influences disease transmission. So factors like rainfall patterns, temperature patterns, long-term temperature patterns, animal migrations, all these things, fire scars, like I mentioned earlier, all affects these dynamics. So understanding these dynamics is essential to disease management. We know from recent history that disease often occur in these clusters, and they follow clear patterns of spread. And if we're sampling these diseases, the sampling locations are not necessarily independent of each other. So therefore, it is important to, for the accuracy of these models and our predictions of our disease models, it is important to include, please consider spatial autocorrelation. So this leads me to my questions, where we investigated the influence of spatial autocorrelation on patterns of disease, immunity, and nutrition within the landscape. And we use various buffalo health and disease metrics to ask the following questions. So firstly, we wanted to know, is there spatial heterogeneity in these disease occurrence after accounting for known factors that we know influences these disease abundance or intensity? Secondly, we want to know, are there seasonal shifts in these disease patterns that we can detect? And then lastly, to what extent do spatial disease patterns reflect heterogeneity in buffalo immunity and nutrition? So basically, if we have these disease patterns, how do they correlate with immunity and nutrition to kind of like help us understand how animals can navigate this landscape of risk? So to address this, I use a relatively new statistical approach called integrated lab-based approximation, and it's an in-lab package called inline-NR. So it's a linear model package with a Bayesian approach, and it's really gaining popularity for the analysis of spatial data. It's especially useful in this context as it allows you to include both spatial and temporal effects within your models in quite an easy way that we weren't really able to do in the past. So to do this, we ran 5 generalized linear mixed models. So we started at a base model, and we increased complexity as we moved forward. So we started with a base model, we included spatial random effects, and then we included temporal effects. And each time we assessed whether that improved the model fit. Okay, so we modeled 35 response variables. So there's 35 times 5 models in total. And so we looked at 5 environmentally transmitted parasites. So that was like the coccidiosis, the cystosomes, the ticks that I discussed earlier. We had 8 vector transmitted parasites. So those were basically Theileria and Anaplasma that gets transmitted by ticks. We had 7 contact transmitted parasites. That was these I mentioned earlier, like Herpes tuberculosis, brucellosis, parainfluenza virus. So contact transmitted ones. 10 immune measures, as I mentioned before, and then the 4 nutritional variables that I mentioned before as well. Okay, so we modeled this with these 5 or 6 fixed effects in each model to account for the variability. So these are things that we know affects the response variables in these models. This was just in there to account for the experimental design in the project, we're specifically interested in that in this case. And all models had an animal random effect to account for the repeated samples. Okay, so INLA allows us, as I mentioned, just briefly now, for inclusion of a spatial random term in model through the specification of a spatially distributed random effect. So INLA utilizes this in stochastic partial differential equations, so SPDE approach to approximate a continuous spatial field using a triangulated mesh of connected discrete locations. That was a mouthful, right? Okay, but basically what it is we want to have a continuous spatial field to estimate our disease, whatever parameter we're interested in. But when we sample data, we sample it in discrete locations. So we want to get from these discrete locations to a smooth spatial field. Okay, so what we do basically is we specify this triangular mesh, and we use that mesh basically estimate the smooth field that we have. Okay. So we included that SPDE random effect into the model to account for spatial autocorrelation in these models. Okay. So this here is what the mesh looked like that we used for this analysis. So we had these 10 sub-bar full of Earth. These are the sampling locations that we use for all of them. And you can see here the sampling locations over here, and we then estimate, use that to estimate the spatial field. Okay, so there's an example of what that will look like. So this is for bovine diarrheal virus, for instance. This is a nice example. So for models that had the spatial random effects, I'm not going into details here, but that's models 3 to 5. That was selected and they proved the model fit. We then visualize these random spatial fields. And when I show you the results, you can think of these as representing the variability in the response variable across the geographic space after accounting for all the covariates, so all these 6 factors that we left here. So basically the variability is left unexplained if you did not account for the random spatial effects. And that gives you a nice spatial view of your data set after you've accounted for all your fixed effects. What's also really nice about these models is from these models you can obtain estimates of the range parameter, or in the modeling language they call it kappa, for each spatial field to quantify the geographic distance over which autocorrelation dissipates. So it's basically, if I have a sampling point over here and I move further away, how correlated are those? And this is really useful when you want to think about the dynamics that your disease operates around, or when you want to think about how you sample in the landscape. So here's just 2 brief examples that I pulled out. I can't go into detail at this time. But here's environmentally transmitted diseases, and you can see here that they have quite a lot of variance between the different parasites. And if you look, for instance, here at environmentally transmitted parasite, much less variance within those variables. So this translates back onto your spatial random fields as well. So high kappa values would indicate smoother autocorrelation. So you can see here that translates to more of a landscape pattern. In the landscape, versus if you have a shorter range of estimates, you have more of these clustered correlations, which you can see here in this diarrhea estimate example over here. We have kind of these hot and cold spots in the landscape. So when you think about the dynamics of the disease, or you think about when you want to manage this disease, this really gives you nice information about the scale that this disease operates. So going back to our first question, is there spatial heterogeneity in disease occurrence? And yes, due to time constraints, I'm just going to show you these examples. But 28 of the 35 models that we ran were improved by including a spatial random effect. So basically, the majority of the models, we did not account for spatial autocorrelation. We left variability unexplained in our models. And that's basically throwing away hard-earned sampling effort. The second question, are there seasonal shifts in these spatial patterns? Here yet again, yes, 16 of the 35 models were improved by also including spatial temporal effect. So the other models we started with just a spatial effect, and then we added season onto that, so it became a spatial temporal model, and 16 models were improved by that. And here you can see some examples here. Yes, for instance, fecal chlorophyll over here. So the overall panel is where we just have spatial random effects, and the dry season, wet season ones are where we include spatial temporal effects. And you can see that there's clear shifts in the estimated fields over there. So basically, we can explain more of the pattern that's occurring in the landscape. So this can give you nice detailed information that you might not otherwise have been able to see and give you insights into the dynamics of your parameters. And the last one, to what extent do spatial disease patterns affect heterogeneity in buffer immunity and nutrition? So here again, I don't have enough time to go into this, but I ran a supplementary analysis where I basically looked at all these spatial random fields for the parasites where the models were improved by including the spatial random effect. And I then correlated that with the immune variables and the nutritional variables, and there was a nice correlation between those. So basically, the immunity variables and especially nutritional variables intersected. And that really can give you nice information about the nutritional landscape and the risk landscape and how animals might need to navigate. Okay, so in conclusion here, so after accounting for variables known to affect the parasite's susceptibility and burden, we are able to demonstrate patterns of spatial heterogeneity in disease occurrence, as well as seasonal shifts. incorporating spatial random effects and accounting for spatial correlation in wild disease research can reveal insights into the factors that drive disease emergence, transmissions, and persistence. And as I said, it can give you a new insight into your data and give you a nuanced view. And we show how neglecting to include spatial temporal patterns can potentially lead to spurious results or inaccurate predictions and ineffective disease management control strategies, which you don't necessarily need to make because we have these new techniques available. So this method of analysis provides an exploratory tool as well for understanding spatial disease patterns and disease risk in a landscape. And lastly, the different spatial patterns demonstrated by the various pathogens and parasites in our buffalo population indicate how variable areas within a particular space can be in terms of these exposure and infection risk. And like I said, the trade-off that animals need to navigate in terms of nutrition versus avoiding parasites. So that leads us nicely into the next chapter, where we're going to be looking at the environment, heterogeneity in resource access, how that plays out in terms of life history outcomes for African buffalo. On time. Access to quality nutrition can have a dominating influence on a herbivore's nutritional state and subsequent fitness and or life history outcomes. So this is all driven by things like foraging ability, animals' competitive ability, like I mentioned in the herd setting, and then in like quite relatively newly emerging fields in terms of personality research, things like boldness or risk-taking behavior can really affect the nutrition that an animal is able to access. Zooming in on the herd structure again, herd social structure dictates an individual's position within the herd and ultimately affects access of good quality forage to that particular individual. Like I said, within a particular herd, there's great diversity of nutritional ability based due to your social spatial position within the herd, with great inequality in both quantity and quantity. So I mentioned the structure over here. So yeah, this was seminal work was done by Prince in the 80s. And published in the 90s. So on the x-axis here is position within the herd. So it's the same orientation as you have over here. You have the front of the herd, and as you move through the herd to the back of the herd. And you can see here on the y-axis is bites per step. So animals in the front of the herd, for each step that they take while the herd is moving and foraging, those animals get to take a lot more bites. And those are generally the good bits as well, as they're the first to get to be good to the forage. So they kind of get first pick and they get to take more of that as well. And then this translates into the second panel over here, body condition on the y-axis and the position within the herd yet again. You can see the animals in the front of the herd has a significantly better body condition than animals to the back of the herd. So the social spatial dynamics really affects the nutritional condition of individual animals, although they're foraging in the same landscape. So this leads us to our questions. So I really wanted to understand the nutritional profiles of these animals a little better. So how do nutritional profiles differ among individuals? The second question, how do individual nutritional profiles affect life history outcomes? And then lastly, are there differential effects and changes of these various parameters and how do they affect, how do they differ across the distribution of these nutritional quality metrics? So for each individual, I had 7, 8, or 9 fecal chlorophyll samples, and I basically just plotted them for each individual and created a distribution. So in this case, I just plotted the mean and the variance for that, and I recognized that there's this very stark, should show you there, the stark non-constant variance viscosity in this data set. And this is pretty typical of these types of data set. But what's really interesting to me then, that led me to ask the next question, which I'll get to now, is that if you say just take one mean of 0.4, for instance, so there's a wide range of ways that an animal can achieve that nutritional outcome of say for a mean of 0.4. So I kind of wanted to understand that a little bit more. So basically, just to explain what I'm going to talk about just now. So animals can have a low nutritional ability. So say for instance a fecal chlorophyll mean of 0.2, or they can have a mean of say for instance 0.6. But how the distribution of all the samples that I have is distributed around that mean was really different amongst animals. So some animals had a distribution that's more like a normal shaped, like almost like a bell-shaped curve, where their mean is really representative of the values that they generally get. Okay. But then other animals had this type of skew distributions, right? So some of them, you know, at the bulk of the nutritional profile was like over here, for instance, but then they had all these outlying values. So they were right skewed. So that allows them to basically pull their mean over by incorporating these bouts of good access to forage quality. And then you had other animals that kind of had the reverse of this, where they generally had high quality forage, but then that was dispersed with like periods where they really had terrible forage as well. So you had either these right skewed or left skewed distributions, or very low skewed distributions in the landscape. Okay, so then that led me to calculate quantitatively the skewness of each individual's fecal chlorophyll distribution, and then that included that in the model. And that's what these lines are on the figure over here. It's just the different amounts of skewness to the longer tails in that. And that mapped really well to the heteroscedasticity that I saw in the status. Okay. So basically, if we take that back to the biology of the animal, so each animal has a nutritional profile, but animals can incorporate various amounts of risk to achieve the same outcomes. So some animals can be really dominant, and they can feed consistently on good stuff. Other animals might be subdominant, and they have to incorporate some form of risk. So they have to change something about their behavior to get access to other good stuff to augment their bad quality diet in general. Okay, so that's basically what that's all. So basically use these two metrics to go forward and look at life history outcomes. So next up, we looked at how do individual nutritional profiles affect life history outcomes. So basically just ran some generalized linear models to look at the probability of mortality and the probability of dispersal. So here we included fecal chlorophyll mean and skewness, as well as I was really interested in that interaction between your average foraging ability and the risk that you had to take to get to that output. Okay, so that's why we then included this interaction term as well. Okay, so let's look at some results. Let's start at panel B over here. So on the x-axis, there is fecal chlorophyll mean value. On the y-axis, the probability of mortality and dispersal on this panel C as well. The lines represent skewness. So the mean skewness in this population was 0.43, and then that's one standard deviation below the mean and above the mean. So let's look at the low skewness values here. So for individuals with a low mean and low skewness, that has elevated probability of mortality. So this makes sense if you think of it in biological terms. So it's an animal that's not eating very well, and it's consistently not eating very well. Now it makes sense that that animal will have a higher mortality probability. So if you then look at as skewed increases for the same mean by increasing the skewness by incorporating some risks, going out of the herd context, for instance, to access nutrition outside of the herd context, or maybe forming a small group and leaving the herd for a certain amount of time, you can reduce that mortality risk by pulling up your mean. Okay, so we then look at animals as they move on here and they eat better and better food. So animals that eat good and do so reliably reduce their mortality risk substantially. But animals that eat good food and does not do that reliably, so those are those animals that I showed you with the left skew distributions. They will basically they face like a hit or miss or a bust or great times stuff scenario where they either can do really well, or if the environment changes and it's all of a sudden a drought, those animals will die. And you can see that is not the best strategy, as it results in a much higher mortality risk. So if you then look at dispersal, and I really think that the dispersal behavior and the nutritional, let's call it strategies that animals need to employ go hand in hand. So if you look at probability of dispersal here, so animals that are eating a poor quality diet and doing so consistently have a lower probability of dispersal. So if you think about biology yet again, these animals are probably subdominant, they're in the herd, but they're probably animals with low risk-taking behavior, low boldness, so they're not going to venture out and do something about it. Okay, so they're not going to come in contact with other herd members or members of other herds, and which will result in lower dispersal probability. As you then go up and they increase their foraging ability, and they do so consistently, you can see that their dispersal probability goes up substantially. So this will be, if you think back to biology again, these will be animals that form a fission group. And by the way, the African buffalo are really known for their fission dynamics, especially when environmental conditions get poor. So when the environmental conditions are good, they will generally congregate larger herds. When environmental conditions get worse, they'll break up into smaller familial units and concentrate in like smaller grazing patches and things like that. Okay, so my theory here and it is of course very speculative, this will obviously need to be tested, is that this behavior allows them to break away from the herd, and maybe other animals from other herds, having the same type of behavior, increasing their contact probability and thereby increasing their dispersal probability. Okay, then the last question I want you to know, are there different effects of different nutritional or foraging abilities? So for this question, we employed quantile regression to evaluate the association of fecal chlorophyll at the different quantiles 0.1 all the way through to 0.9 using the LQMM package. And we included all these variables to account for them. But what I was really interested in, is to see the effects on mortality and dispersal. So these are all the parameters we included in the model. And I'll show you the results of that just now. So I'm not, there's a lot of interesting results here, but I'm kind of just going to glance over this just to tell you that there was differential effects across the quantiles, so across the nutritional ability of these animals when the environment changes or when the body condition changes or when they age. But what I really want to focus on now, just in terms of time constraints, is the mortality and the dispersal results yet again. So I was interested to see if you're eating poor quality food, you're eating good quality food, and we know that you died, was there a different effect in terms of what we predicted that you're going to eat? And what was really interesting to me, if you look at the mortality intervals, so these are the normal intervals we sampled you before you died, and this is the light gray here, you can see that the confidence interval there overlaps with 0. So animals that we know died was not predicted to eat any different from animals that we know didn't die in this study. And what was interesting is that was across quantiles as well. But very interestingly, when these animals did die, so the dark gray area, they were predicted to have much lower fecal chlorophyll, so much lower nutritional ability or foraging ability. And yet again, that was across quantiles. So that was interesting to me as well. So I was expecting animals with the higher quantiles, to maybe not be as affected by this. So that was pretty interesting result. So it doesn't matter what your nutritional or foraging ability, if there's something that happens in the landscape that perturbs the landscape and your nutritional ability drops, that will increase your risk of mortality. If we then again look at dispersal, kind of wanted to see here again, is there anything that I can pick up across the quantiles that's characteristic of dispersing animals? And interestingly here, yes, there was an effect. So if you look at the light gray shading again, those are animals that we know dispersed, and over the normal intervals, they actually performed and were predicted by the model to have much lower fecal chlorophyll values. So the foraging ability is much lower than average individuals that we know did not disperse in these studies. And then if you look at the interval that they did disperse, these animals were predicted to have a much increased fecal chlorophyll value away from the norm and higher than animals that we that did not disperse. So yet again, it kind of speaks to the results we had before. These animals, when they disperse, there's something different that happens in their nutritional and their foraging behavior and ability that increases their probability of dispersal. Okay, so that takes us to the last chapter. We're going to zoom into a specific parasite, Schistosoma matei, and we're going to look at how animals resist infection by schistosomes and how this investment in resistance plays out in terms of life history effects. So infections are ubiquitous in wildlife, and especially because, as I mentioned in the introduction, a lot of these parasites and pathogens become endemic in these wildlife systems. So animals can't simply, get rid of them. Not like us who get flu, we get the flu, we get over it, and we don't get it for a couple of years again. So animals have to really deal with consistent onslaught and consistent burdens of parasites. So there's two ways that they can deal with this. They can either actively fight these infections through resistance mechanisms, or they can deal with it through tolerance. So basically this grinning and bearing and managing the effects of those parasites. Today, I'm going to focus more on the resistance aspect, exploring characterized as the host ability to limit parasite burden, so i.e. the number of parasites infecting that individual host. So parasite exists at the expense of their host. So one logically might expect that the trait enhancing resistance to evolve through fixation. Animals that's basically excluding parasites, right? That would make sense. But hosts, we don't really see this, because hosts often suffer differentially when they're exposed to infectious agents. So there's great variability in host susceptibility, parasite infectiousness, the length of the infectious period, infection intensity. And this suggests that variation in resistance is maintained in these populations. So if resistance did evolve to fixation, then parasites would quickly go extinct, as I mentioned. So this is really where we, as ecologists and evolution biologists, are really captivated by the evolution of resistance, and specifically how does this maintain and how this variation is maintained in these populations. The key to understanding this, the key concepts to always keep in back in mind, is that the maintenance of resistance is costly in terms of, for instance, immunopathology, and it's also energetically expensive, and especially when the environment changes and parasite pressure changes, and if parasites get excluded from the system, when parasite pressure gets reduced, these are potentially damaging to the host as well in absence of those infections. So one mechanism through which these resistance dynamics and tolerance dynamics might be maintained is negative frequency dependent selection. So this occurs when the frequency of resistant phenotypes cycles asynchronously with parasite prevalence. So when resistant phenotypes are common, there's reduced parasite exposure risk, and then that in turn relaxes the selective pressure on resistance, favoring non-resistant phenotypes. And then as we progress, as the host population shifts towards this higher frequency of non-resistant phenotypes, it flips around again and the parasite can once again spread throughout the host population, increasing selective pressure on resistance once again. So you have this basically ebb and flow of the parasite winning and the host winning, and how they modulate their resistance phenotypes within the landscape. So, although much work has been done on this particular parasite resistance dynamics, it's mostly been done in invertebrate systems. And we actually have a few studies that document selection, resistance, and maintenance of this variation resistance in wild terrestrial vertebrates. So we have a few lab examples of this, but in the wildlife setting, there's only been a few studies that's been done. And this is due in part to the challenge of identifying resistant phenotypes, and especially where they're supported by genetic analysis within these unmanaged populations. So the ability to measure these fitness costs and the benefits of resistance is really hard. So in this study system, we were lucky that we kind of had the opportunity to do this and address, make a little bit of a dent in some of these questions. So we tracked, we were able to track schistoderm burdens using a titer called schistosome-specific circulating hematotic antigen, and CA titer for short, in 196 buffalo. So basically, the titer just estimates the burden of schistosomes within a specific animal. So that allowed us to ask if schistosome burden is a heritable trait in these populations. So we need to do that to determine if this trait can undergo selection and whether these mechanisms I just discussed can actually be active in this population. And then secondly, is there variation in schistosome resistance amongst individual hosts? So we know that the burden increases over time. So do some animals reduce that burden accumulation, or some animals can accumulate it faster? And then are there fitness outcomes associated with schistosome resistance? So just very briefly, quickly discuss the life cycle of schistosomes. The schistosomes require this molluscan hosts, and so this makes the infection dynamics of this species really spatially dependent. And later on, I'll show you we accounted for this by also using inline models for this analysis. So the molluscan host basically secretes cercaria into the aquatic environment. So these molluscan hosts are very dependent on wetland and river type habitats, and the infection dynamics of this is highly driven by where they are in the landscape and how animals actually enter and utilize these aquatic landscapes. So if cercaria gets secreted into the water, they get secreted by the thousands all the time. They're very localized around the molluscan host that secreted them. If an animal then ventures into the water, they penetrate the host and they mature and breed in the host, and then eventually Myricidia is secreted back into the water, which then reinfects the molluscan host, and the cycle starts anew. So we used single snip techniques basically to estimate heritability, whereby we calculated the relationship between animals, so how genetically related animals were from each other. And when we use this information to determine pairwise relatedness amongst buffalo, we incorporate these pairwise relatedness estimates in a matrix as a correlation structure in a random effect model. We used repeated measure variance components models to estimate the additive genetic contribution to variance in schistosome burden over time as individuals age. And we could then calculate the heritability estimate from that. So here's the result. So each schistosome burden, a heritable trait. Our estimate for this was 0.446. And this means that this is a highly heritable trait in African buffalo. Okay, so this may be a bit of an abstract number if you're not familiar with these types of traits. So here is just some comparisons. So here is schistosome mansonii, and these are just human examples. This one is in Brazil, an estimate of 0.41, so very close to our estimate. The Japhonicum from China, 0.58, a little bit higher. And then if we look at some wildlife examples of nematodes and strongbill worms, we see much lower estimates in blackface sheep, 0.14, and soy sheep, which is another very charismatic wildlife example, of 0.26. So just to put that into context. So moving on to the second question, is there variation in schistosome resistance amongst individual buffalo? So from previous work that was done by some of my colleagues, we know that schistosome burden increases as animals age. So generally, schistosomes will cycle with seasons and they'll go up and down, but as animals age, their burden will slowly increase. Okay? And some animals will do that faster, and some animals will do that slower. And we can see this with the increased variability as animals age. So some animals will do that faster, the variability, and as they age, the variability will increase. So animals are doing different things as they age. So we wanted to basically look at this and see if we could estimate individual schistosome burden growth trajectories. So how we did this is we basically just used a linear mixed model, and we specified age as a random effect with random slopes. So from this, we could extract basically individual slope estimates for each individual animal. So for instance, animal X, would have a low slope, meaning that animal is more resistant and its burden accumulation is slower, whereas this animal with a high slope estimate, so positive value for the slope estimate, where that animal is less resistant and accumulates burden much faster over its lifetime. So that's the simplified model formula for that. And then we also included those SPDE random effects to account for the spatial location and the spatial dynamics of the molluscan host as well, and the variable infection dynamics that will cause. So here was our sampling locations for this. So in this case, we used the two main herds as the herd variable. And then there's the mesh that we used to generate the spatial random effects as well. It's similar to what I discussed in the first chapter. So here is the spatial random effects. So from this, we could see that there was a bit of a seasonal effect, which previous work has also seen, a bit of higher schistosome burden in the dry season versus the wet season, and generally much higher burdens in the south, and it gets reduced as you go northwards in the park. Not exactly sure, there needs to be some research there why you have these higher burdens in the south, but there's quite a lot of preferable aquatic environments in the southern area, which is definitely more favorable to the molluscan host than, for instance, in the north. So is there variation in schistosome resistance amongst individuals? So we also found the same age and burden growth of age in this population. So this is what graph A shows us here. So it's just age and the log of the titer. And we saw this positive slope of the general population estimate. So we looked at panel B here. This is our extracted slope estimate. So there's random slope estimates for each individual. And that is just the distribution of that. So we can see there that there's quite a bit of variability in the slope estimates. So meaning that animals have different levels of resistance to schistosomes. So just as a bit of a key here, so the dark bars here is lower slopes. So those animals will accumulate schistosomes at a slower rate versus the non-resistant animals with the light bar there, that will accumulate schistosomes at a faster rate. So that moves us on to our third question. Are there fitness outcomes associated with schistosome resistance? So individual schistosome burden growth trajectories provided us with this continuous estimate of trait variation in schistosome resistance after we controlled for those various things like age and herd membership and the spatial effects. So the effects of schistosome resistance on, so we used this then to look at the effects of cystosome resistance on reproductive outcomes. So how animals bred, their somatic growth, so basically the horn with the investment in that secondary sexual trait, and then also survival over each capture interval. So if we just look at the reproductive output, we again specified a generalized linear mixed effect model. Here's the simplified model formula over there. So those are the variables that we included. We included the interaction term between cystosome resistance and age as well. So here's the result. So we found strong interactions between cystosome resistance and age on the probability of detecting a pregnancy, but this was variable depending on the age of the buffer. So here we have the cystosome resistance that's depicted in the tile strip over here, and in age, the different lines that you see. So 0 is the median age, that's 77 months. One standard deviation below and above. So the solid black line is one standard deviation below, so young animals. So you can see here, when they mount a resistance response against schistosomes, they have a much higher pregnancy probability. This is animals that did not mount a resistance response. This, however, gets flipped around when you look at older animals, and the effect changes. So here, animals that have more of a tolerance or less of a resistance response has much higher probability of pregnancy for older animals. So secondly, we looked at somatic growth. So here's been a very similar model framework, exact same parameters to give us this result. So resistance to schistosomes was associated with reduction in horn width, with the effect being more pronounced in older animals. But generally, we have this positive relationship. Okay, so for animals that mount a resistance response against schistosomes have smaller horns in general, and this effect gets amplified as animals age. Okay, so what is this? If you think back to the biology of the animal, what does this mean? So like I discussed in the beginning, resistance phenotypes basically requires something, and nothing is for free in these systems. So mounting an immune response or a schistosome response probably costs energy, and animals have to make a trade-off between investment in this case, the model we're looking at now, somatic growth versus either survival or reproduction, for instance. And then lastly, we looked at survival, very similar model structure. We looked at the probability of mortality, and here's the result of that. So the probability of the per capture mortality is associated with cystosome resistance, and especially this is very strong for the younger age classes. So you can see here, for young animals that mounted a schistosome response, they had much reduced probability of mortality versus animals that did not mount a schistosome response. And then for older animals, this response was a little bit reversed, but not nearly as strong as for these young animals. So what this really, I think, shows me is that for young animals, I think when the parasite resistance, the cystosome pressure in the landscape is really high, this resistance strategy really pays off in terms of juvenile survival. And as we know, for buffalo population dynamics, juvenile survival is really what drives, how much buffalo we have in conservation areas in the long term. So juvenile survival is really important, but it only pays to mount this response to when the parasite pressure is high. So if parasite pressure is not high, then you're just going to be wasting energy, and it's better to then invest in, like we saw in the previous slide, in somatic growth, which is then going to pay off in terms of reproduction later on in life. Okay, but that doesn't help you didn't survive in the first place to get to that. Okay. Now I went from script here a bit. So both cystosome resistance strategies in our study population seemingly have fitness benefits, suggesting a reason why resistance is heritable despite having all these different fitness costs. And the results here suggest that investment in cystosome resistance comes with energetic costs that need to be balanced with lifetime reproductive output and survival, and not all interacts with cystosome pressure in the landscape, with both these strategies being maintained in the landscape as cystosome pressure in the landscape cycles. Okay, so that takes us, that was the last chapter. So just to conclude, we first started off with parasite distributions and exposure risk in the landscape as the first leg of our epitriad. And then we demonstrated that spatial and temporal distributions of disease risk within the landscape. We demonstrated these spatial patterns, including seasonal effects, and we showed that there was correlation between parasite spatial distributions as well as immunity and nutritional distributions. Secondly, we looked at the environment and variability and access to nutrition. We showed that access to quality nutrition has consequential life history outcomes in terms of reproduction and survival using this novel dietary metric, fecal chlorophyll. And then lastly, we zoomed into a specific example in the case study of Schistosoma matei, where we showed resistance to schistosomes is heritable in African buffalo, and we have disparate fitness effects, possibly providing fitness benefits when parasite pressure is high and conditions and environment is low, resources are low. So these results suggest a mechanism for the maintenance of these two disparate resistance tolerance strategies. That concludes this whirlwind tour. I just want to say thank you to my committee, Anna, Bree, Michelle, Ben, and Lisa, for your support during this whole endeavor. Your feedback during the prelims and things like that, I really, really appreciate all of that. All members of the Johnny's Lab, first, a lot of them are graduated already, so past and present. Thank you for your friendship, support, feedback during lab meetings, all of those things. Then to Devin, Lindsay, and Laurie, which throughout my 6 years at OSU provided me with a job and a way to live. And thank you for all your conversations and everything as well. And also, really importantly, always thank you for always being considerate of and accommodating me with the family. That was always greatly appreciated as well. Then just everyone that was in the field, I mentioned them before. I've used a lot of people's data sets, including my own. So that was great. Thank you for your hard work. Sandbox Veterinary and Wildlife Services. Without we'd never be able to dart and capture these animals. Thank you there, especially these bus, which is head of Veterinary Wildlife Services. And then last but not least, the IB staff for all their support during my time here at OSU as well. And then lastly, my screen didn't change. To my long-suffering family, my wife Lee, we literally, without her continuous support and motivation throughout this whole process, I would not be standing in front of you here today. And then to my two daughters, Kenna and Kai there, thank you for just being troopers and playing outside when I need to work on Saturday, keeping yourselves busy. Thank you, and hopefully I can spend more time with you now. So thank you, everyone, and I'll take some questions if there are any.
Her mother wants to know when the 4 Dummies version is coming out.
The 4 Dummies. It was hard to condense all of that down into 15 minutes. Yes.
All right, so you have a SNP that's associated with what you're terming resistance. Schistosomes have been around for a very long time in the study system, and it seems like this is a very strong correlation with it. Why hasn't it just become the absolute genotype throughout this population? What requires it? Why is the other SNP even hanging around if this schistosome is such a problem? It seems like there should have been such a huge selective pressure on this that it should just be homogeneous by now.
So the SNPs were not for schistosome resistance, the SNPs were for relatedness in the population. So we use the relatedness estimates as the various structure to determine the broad sense heritability. But I can answer that question. So a lot of these other diseases that's introduced into the system, like TB and foot-and-mouth disease, are new diseases. So we expect them to have these really strong resistance dynamics because they're still evolving and developing. So this was kind of why it was interesting to look at this disease, because this is an endemic disease that's been in the system for a long time. So you would not expect these strong resistance effects, right? Because there's been a long evolution history there. But yet again, we see that these strong disparate dynamics going on here. So that was really interesting. So over a long evolutionary time period, resistance are being maintained and divergent strategies are being maintained within this population, which I think was really, really interesting. And a lot of it's speculation, just knowing the ecology of these animals, but we think that these two strategies where juvenile survival and reproduction is being prioritized when the parasite pressure is high versus when the parasite pressure is low, then animals will delay reproduction, invest in somatic growth, and not invest in resistance, and then that will pay off later in life, because you can make it through with parasite pressure. So because because there's such strong seasonal dynamics and spatial dynamics in the system, I think that allows these divergent strategies to be maintained over these long evolution time periods.
So as far as parasite pressure goes, is it, I guess, most closely correlated with seasonal changes, or like what is the time frame as far as the parasitic pressure being high to low, and then the resistant, non-resistant phenotypes being show up.
So, so there's seasonal like parasite pressures, like as I mentioned, the dry season parasite pressure will go up, and this is just due to the ecology. So this system dries up, as I mentioned, in the dry season. So animals will congregate on these wetland and river type habitats where you do get the snail hosts, right? So they don't come in contact. So that's when the burden will go up. It probably also cycles with just general like immune ability and nutritional condition of these animals as well. The immune system is probably suppressed in those times as well. So that probably also elevates the burden as well. So that's like short-term cycles, right? And then you have these longer-term climatic conditions, which is driven by like El Nino cycles and things like that. So that probably influences that as well. So I suspect like maybe during drier cycles, the pressure is going to go up just due to animals congregating on like wetland type of habitats. Where it's more wet, I think parasite pressure is going to be a little bit lower. But the people in the room might strongly disagree with me on that. I'm not 100% sure. And then you just have this spatial diversity, which that's kind of why I highlighted that in the beginning. So I think just over space, you have different dynamics going on as well. So in one location, the system pressure might be really high, in another location, it might be really low. And then if an animal migrates to a different area, different phenotypes are going to be selected in the same geographic space.
As far as mortality goes, what I caught was that it's treated as sort of a uniform factor. Do you think, well, I guess, do you know what is the variety of actual causes of mortality? How do they end? Are they all consumed by lions? Does this matter in terms of your analysis? Do you think it affected anything?
I mean, it definitely does matter. So buffalo, when they stay in the herd context, are really safe. So predation within a herd is really low. They protect each other, protect calves within the herd. The tricky part comes when you need to separate from the herd. So either you are pregnant and have to give birth, or you are doing nutritionally very poor in that herd and you need to separate from the herd so you don't die within the herd. Right? That's when predation pressure really starts coming to play. And that's what I think we see in some of my results, where when these animals that have these disparate nutritional profiles, incorporating a lot more risk, and that risk is probably predation risk, because they're moving, they're separating themselves through either just breaking away in small herds or breaking away individually, and that's when they get nailed by things like lions. And then obviously there's disease deaths as well. So TB, although it doesn't kill animals over the short term, they definitely die of TB-related complications as well. And then there's a host of other things that, and a lot that we don't even know about, that also cause mortality. And then sometimes they just die being stupid, getting stuck in the mud or something.
Yeah, so you have that sort of age switching factor for pregnancy and mortality. How much might size itself be a factor, as in, can a younger animal sort of get swamped by Schistosomiasis itself?
I'm not sure. Yeah, I don't know. I mean, if you think about maybe a big animal might have more resources that might support higher burdens. But if you think of it, like where they are in the Mesogeic or in the hepatic veins and stuff like that, I think there's only a limited amount of space that they can hold in. Yeah, I'm not 100% sure.
If you had the chance to follow up on one burning question that arises in the study, and you could do stuff on wild buffalo to answer that follow-up question, what would it be?
Well, you know, I think this system, and you're kind of addressing that in your new study that you're doing now, is just if we had collars on each animal, contact collars, and so we could understand how animals interact socially, how they contact each other, and then how they move in space. So getting a better grip of like the fine scale of spatial dynamics. So this one shortcoming of this study is for mine at least, was that we had 6 monthly time intervals, and a lot can happen over a 6 monthly time, right? And it's just a little tiny snapshot of what's happening in that animal's life. So that's what we have, and that's what we work with, because these difficult or data are difficult to get. But if I can do one thing, yeah, I will increase sampling intensity and include like spatial dynamics, more frequent temporal samples of spatial dynamics. Yeah, and then also maybe just sample the animals throughout their life. So from
That would be
birth
great.
to death.
That would be
Put a
great.
collar on every buffalo and cougar.
Yeah, that would be great.
Sample for their entire life.
Yes.
So much for like an
Just like we need to keep looking.
So I think there was a lot, especially the mortality dynamics that we saw, that I think if we monitor these animals later in life, I think so, especially for cisplatin-resistant, for instance, I think that the effects of that and lifetime reproductive outcomes and stuff like that would have been very interesting to see.
Can I suggest you switch to invertebrates?
Yes.
More questions for Henry. Right, well, then I guess we'll wrap up this public portion of the defense and wonderful talk, Henry. Thank you. Also, for anybody who's not on